Good afternoon. So today you all know is the Ides of March. So all Shakespeareans know that that's when Julius Caesar met his end. Not that we're contemplating anything like that today, but I figured I ought to say something about it. At any rate. Okay, so there are a few people who haven't been here before, so I should just very quickly remind you what this is. Do you know what this is? What is it? Huh? What is it? Anybody? It's the Brooklyn Bridge. So it's the most famous bridge in the world. So why do we show the Brooklyn Bridge? Because it symbolizes what the purpose of this whole course is, which is to deal with what many of us think is the biggest problem in science today, which is not new technology, and it isn't even new drugs, it's communication. It's the great advances in basic biological engineering sciences, which you can make believe are taking place on that side of the river, which is Manhattan. And then there's clinical medicine, patients, disease, and so forth, which is on this side of the bridge, which is Brooklyn. And the real name of the game is how to get people to talk together. The other interesting thing, of course, is once you build a bridge, life is never the same on either side. So what we try to do is to combine in this program, usually when we can, a live patient, who those of you here last week will recall, who in 5 or 10 minutes describes what it's like to have the disease they have, puts a human face on a disease, as someone put it. And then someone who's a physician, clinical, translational investigator who talks to mainly basic scientists, who are most of the audience, about fundamental things that you should know about the disease, things that you can readily understand without having taken a specialized course, things like epidemiology. How do you know whether somebody has it? Where do we stand to diagnose? And then why that person is here at the NIH spending their career doing exciting stuff. And what are the challenges? And there's plenty of room for questions, comments, and discussion, and we encourage you, particularly if medical jargon starts flying around and you don't understand, say, hey, wait a minute, and we'll start over again. And then the second speaker is someone who's more, as a basic scientist, dealing with fundamental mechanisms that are involved in some aspects of it. Now, today we have a special treat because I'm going to start by introducing the second speaker first. So the second speaker is Rosana Capeller, who graduated from medical school in her native Brazil, and then received her PhD degree in the department I was the chairman of at Tufts in Boston, molecular cellular physiology. And she trained with Lou Cantley and a lot of very exciting people. And then Rosana, after a postdoctoral fellowship at Dana-Farber, got interested in another aspect, and that is, I don't want to call it the commercial, but that's really what it is. It's the explosions that were taking place with biotech companies being formed and dealing with new products, new ideas, venture capitalists, and so forth. And Rosana is one of the most successful and energetic people in bridging this field. And so this is a different kind of bridging. This is a top-notch basic scientist, physician, who was also an incredibly accomplished, I don't want to use the word administrator, but at any rate, I guess that's what it is. She's the chief scientific officer of Nimbus Therapeutics, which is a small biotech company in Boston. And it's on the basis of work and projects going on there that she has developed quite an expertise in the topic of today. And so our first speaker is going to be Jeroen Rotman, who's chief of the Liver and Energy Metabolism Unit in the Liver Disease Branch at NIDDK. So he graduated from Hebrew University at Hadassah. He trained in gastroenterology at the Rabin Medical Center in Petah Tikva, came here for a hepatology fellowship, and since August of 2009 has been a faculty member, a tenure-track investigator. In the beginning, he focused a lot on hepatitis C, but that's tending to become a bit passé. And several years ago, he shifted his main interest to the pathophysiology of fatty liver disease. He's involved in clinical trials. He's involved in collaborative studies of various kinds. And if I can get this thing to work. Okay, so here are some of the quick things that went through my head when we put this program together. And you probably have your own, but just to give us a little focus. So one of the curiosity is, prior to 1990, nobody talked about non-alcoholic fatty liver disease. In fact, if one had a patient where on a biopsy there was fat triglycerides sitting there, you would interrogate the hell out of the patient, accusing them of alcoholism, being a closet alcoholic, because all we knew was alcoholic liver disease. Now, there were a few rare metabolic disorders, porphyria, occasionally hemochromatosis, a few other things, diabetes, where there was some lipid, but that was pretty rare stuff. If you had fat in the liver, you were an alcoholic, so you better tell me about it. But I don't drink, doctor, I don't drink. And what happened? It's one of the big mysteries. I mean, the current vogue is that the diet changed, the cultural pattern changed, more calories, more carbohydrate, more fat people, more diabetes, more fatty liver. And maybe that is it. That may be an important catalyst, but as you'll hear, it's not the only story. So the next thing is non-alcoholic fatty liver disease, which is alleged now to involve millions of people all around the world, but particularly in countries that have high caloric intake and maybe not so much exercise, I don't know. But, what is it? Is it a syndrome? That means you got fat in the liver, but it can be caused by many different causes. Or is it a specific disease? Is it a defect in XYZ gene? Well, for some people, those are facilitating factors, but for a lot of people, they aren't. Not everybody who has fatty liver disease is fat. Many are rather skinny in certain parts of the world. It's a lot of ups and downs. Next, is non-alcoholic fatty liver disease serious? I mean, does it matter if you have more fat in the liver? We don't really know the full answer to that. But we do know there is something called steatohepatitis, which is an inflammatory, necrotic, fibrotic, cirrhotic, cancer-producing sequence. Now, does NAFL progress to NASH? Epidemiologically, it does somewhat, but we don't know. Now, that's important because NASH is a serious business. Leads to transplantation, leads to death. And a lot of times it leads, well, I said to transplantation. So if NAFL is like the part of the iceberg that's below the surface of the water and NASH is what's on top, should everybody with NAFL be treated with drugs if you can develop drugs that affect fatty liver? Now we're talking about hundreds of millions of people and probably hundreds of millions of dollars, which is certainly a catalyst in the sudden burst of interest in NAFL and NASH. I think one of the other reasons for the sudden birth of interest is because hepatologists have now more or less licked hepatitis C, which was the big topic. This is the next big topic. This is touted as the major liver disease increasing. Okay, so how do we diagnose it? Is a liver biopsy good, bad, indifferent? Are there markers where you can just test? Are there non-invasive ways? How do we tell? Anyway, that's the broad subject of today. And you're going to answer all these questions.
Okay, working. Thank you, Win, for this introduction. I feel like I'm on the CNN Choose Your Party debate where I was hit with some questions I wasn't prepared for, but I'll try and answer at least some of them, and the rest I'll just confabulate like the politicians do. So just some disclosures, I don't make any money out of this, but I really am fascinated by the disease. Everything I will say therapeutically is off-label because there is no approved therapy for fatty liver disease. And just as Win said, beware of the Ides of March. But what is little known is that it's because of the liver, because the soothsayer who warned Julius Caesar 2,060 years ago today actually based his recommendation on readings in the liver of slaughtered animals. I'm not sure if they were kosher or not, but something. So if your liver says something to you, you'd better listen to it. I didn't bring a live patient, but I did bring a case, a demonstrative case. So John Doe is a 52-year-old Hispanic that I see in my clinic. He was very incidental. Well, we'll assume we have a pointer. He was incidentally found to have elevated liver enzymes. ALT is the marker that we typically use to detect liver disease. His only symptom was mild fatigue when we really questioned him. This was found just on a routine physical. Physical examination is notable for being obese. His BMI or body mass index is 37. For the non-clinicians, the normal range is 19 to 25. For me to be with a BMI of 37, I would have to gain something like 45 kilos or 100 pounds for my height. So that gives you a range of where he is in terms of obesity. He has other disorders that are associated with obesity, like hypertension, obstructive sleep apnea. We ruled out all the other risk factors for liver disease. We ruled out relevant serologies. We sent him to have an ultrasound. The ultrasound shows fatty liver. So that's what he has. He has non-alcoholic fatty liver disease, or the subject of our talk today. I did something wrong. I did something right. So this is not him, of course, the late Julius Caesar. So this is what I'm going to talk about today. I'm going to talk about some clinical features of the disease, not all of them. I'm going to highlight some aspects of mechanism, and then I'll let that lead us to discussion of treatment options, at least what we have now in the clinical world. But first, and this is a nice connection to what Win said earlier, this is the first description in the literature. It's from 1980 from the great Ludwig in Mayo Clinic. It's worth reading this out loud. The recognition of non-alcoholic steatohepatitis will have immediate benefits for patients and their doctors. The affected patients no longer will be accused directly or by implication of lying about the alcohol intake. And here comes the important part. The physicians will be spared the embarrassment or worse. So truly, this was the first time anyone actually coined the term non-alcoholic steatohepatitis. And if you look at what's been published in PubMed, until the mid-1990s, there was about 1 to 2 papers a year about the topic. Currently, I think we plateaued at about 1,000 papers a year published just on that term. So obviously, there's an explosion of interest, an explosion of knowledge, maybe an explosion of the disease itself. So let's define it. What is non-alcoholic fatty liver disease? This is what it is. It's just excess fat in the liver. And when I'm saying fat, I mean triglycerides, and I'll probably use the terms interchangeably. How much is too much? Well, we have data from 2 different studies. One of them is an old study done at the NIH in 1970, where just autopsies of normal people. And if you look at the data and you calculate it, it's not in the paper, but the 95th percentile is about 50 milligrams per gram, or about 5%. And then we have a population-based study from Dallas using MRI measurements, and they reach the same number, about 5.5%. So 5% or so is considered the normal upper limit for triglycerides in the liver. We can break the disease into 2 different forms. One is just steatosis or blend steatosis, or some people call it NAFL or NAFLD, just fat in the liver, nothing else. The other kind of the other part is non-alcoholic steatohepatitis, which has the itis in it. It has the injury, the inflammation, and this is a progressive disease that eventually can lead to cirrhosis with all of its complications, and can lead to liver-related death and to liver-related suffering. How common is it? So it depends how you look, depends which tool you use, and it also depends when you're asking the question. If you look at data from the late 80s, early 90s, this is based on the NHANES, which is a US epidemiological and nutritional survey. So if you look at that population by elevated liver enzymes as the marker, about 5% had fatty liver. When you look at exactly the same population with imaging, 20% or nearly 20% had fatty liver. And that's a more accurate way to look at it. And if you go a decade and a half or 2 decades later, these are 2 population-based studies from Texas, and you see the numbers range between 30 to 45% of the US population. It's now the most common cause for abnormal liver tests. It's the cause for, if you don't have any other explanation for abnormal liver tests, this is the cause in most cases. It will become the leading cause for liver transplant and for liver-related mortality. If you think it's just the US, no, it's just a problem of the Western world. It's not. This is a nice graph, again, about a decade ago, showing the association between obesity in different countries and prevalence of fatty liver in different countries. And this was just compiled from different papers. But you see, this is happening everywhere. And it's seen in pediatric populations, and kids are presenting with cirrhosis now. So this is not trivial. You think it's far away, it's in Texas, or the rest of the world and not here. This is work that we did, we're working on. We looked at the healthy volunteers to clinical trials here at the NIH. Anyone here is a postdoc, must have been a participant in at least one study. And we just pulled almost 3,500 patients. This is done with Varun Tucker in our group. Almost 3,500 patients who were volunteers here in 5 years. 56% of them did not have normal weight. 45% of them did not have normal liver enzymes. And when you combine the two, just as a very rough marker, 30%. So even if I'm wrong by double the estimate, it's still a huge number of people have fatty liver. And this has a lot of implications. If anyone is recruiting these patients, for example, for drug metabolism studies or other things, this may actually have a lot of impact. So our tool, we are dependent on a liver biopsy. This is Julius Caesar undergoing a liver biopsy or not. And we are dependent on it because this is what tells us really what's going on in the liver. And we're looking at several axes that are not perfectly correlated with each other. We're looking at I'll start pointing to this side so everyone gets their share. We're looking at the amount of fat in the liver. We're looking at the amount of inflammation, the inflammatory cells that infiltrate the liver. We're looking at cellular injury of the hepatocytes themselves. And we're looking at the outcome of all of these, which is the fibrosis, the deposition of collagen. And you can see here in an example, we have the top picture is normal liver. The bottom picture on the left, you can see the white globules are where the fat used to be and was washed out by formaldehyde. There's a sprinkling of inflammatory cells. There are cells here that are ballooned, swollen. distorted. These are cells that suffer from injury. And if we use a blue stain for collagen, we see collagen deposition that can eventually lead to fibrosis, cirrhosis problems. And something that has plagued the field is that we actually do not have any reliable way to differentiate NASH, the progressive problematic disease, from just fat in the liver. There are a lot of studies about biomarkers, serology, imaging. None of them is good enough actually to allow for clinical decision making. Is fatty liver, going to natural history, is fatty liver even a problem? Well, first, does it progress or does it not? So this is a nice study that took people with paired liver biopsies that had more than one liver biopsy, and they showed that if you just had fat on your first biopsy with no inflammation, no other markers of injury, your chances of progressing towards liver disease and developing fibrosis were not very high. On the other hand, if you already had some degree of injury, some degree of inflammation in the liver biopsy, you were very likely to progress, and at 12 years, more than 75 or 80% of these people actually already progressed and developed advanced fibrosis. So there are definitely two populations, one with progressive disease and one without. NAFLD is associated with increased mortality. This is a Scandinavian study that had 30 years of follow-up, and compared to the population in the dark line, the dashed line shows that people with fatty liver disease at the beginning of follow-up actually die more than their age-adjusted colleagues or other people. But when you break it down to NASH and non-NASH, or here they broke it down by fibrosis, but it doesn't really matter, you see that it's the NASH patients that die more, that the others are very close to the general population. And this can be broken down in more detail, but overall, we have a progressive disease that can kill you. What do you die of? Well, this is work that we're doing with Ohad Etzion, that was here, and David Yardeni, who is in Beersheba in Israel. And we pulled data from almost 11,000 people who underwent cardiac perfusion scan. There was a question of whether they have cardiac disease, and they had the nuclear scan to image their heart and its perfusion. And following them for 5 years and looking at those who have elevated ALT with no clear explanation, about 15% or 14%, and we're assuming the majority of these are patients with fatty liver. What's the impact on mortality? It's actually very interesting. They have an increased cardiovascular death. They have a higher risk of cardiac death and a higher risk of developing myocardial infarct, a heart attack. And interestingly, this can only be shown in those that had a normal scan at baseline. So let me interpret it. If you have a scan that shows you have significant heart disease, it doesn't matter what your ALT is. You're already sick enough and you'll die of heart disease, and your ALT doesn't modulate that. But if you actually think you're healthy, you have a scan that shows your heart is reasonably well, but you have elevated ALT as a surrogate for fatty liver, you're actually at risk of progressing and dying from cardiovascular disease. So you think you're mild, you'll progress. And indeed, in other studies, this is data from the Swedish cohort, cardiovascular disease is the #1 cause of mortality for fatty liver patients, the same as in the rest of the population. And cancer is the #2, at the same as the other patients. They just have increased mortality, but they die of the same causes. They can die of liver disease. It's the 3rd cause instead of 11th in the general population, but it's only 6% of them will die of liver disease. So as a hepatologist, when I'm treating NASH, I actually have to think about the cardiovascular health of my patients. Sorry, Caesar. Sorry, I couldn't resist it. I thought it was a good one. Let's talk a little bit about mechanisms. And I'd like to focus on 2 things. We could spend 3 days on talking on mechanisms, but I want to talk about the fate of fat. Where does fat come from and where does it go in the liver? And then we'll talk about some injury mechanisms. So fat can come to the liver in the form of free fatty acids from 3 main sources. There are triglycerides coming from the meal, and this is a very minor component and almost negligible. They can be arriving to the liver through release from the adipose tissue in the form of free fatty acids, and can be generated in the liver as de novo lipogenesis. What can happen to these free fatty acids? Well, they can be converted to triglycerides, which can then be secreted as a VLDL, as a lipoprotein particle. they can also be oxidized, metabolized to predominantly through beta oxidation, although there are other metabolism pathways. Let's look at them one by one. So let's start about the delivery of fat from adipose tissue to the liver. So fat, in essence, is the molecule is an energy storage molecule. It's when we have excess calories, when we're eating, we can store energy. When we're starving, we can use that energy. It's like a capacitator. It keeps the energy supply, can keep it in a stable level. For that reason, when we're feeding, we don't need fat to be released from the adipose tissue. We have calories from the food. When we're fasting, we need the fat to be released from the adipose tissue. So insulin, the hormone of satiety, essentially, the hormone of energy availability, regulates the release of fat from the adipose tissue and regulates lipolysis. And in normal subjects, shown here in black, the release of free fatty acids from adipose tissue is decreased about 80% when you give them insulin. This is the normal reaction. On the other hand, if you look at patients with fatty liver disease, this is actually, it's decreased, but not to the same degree. So what we're seeing here, we're seeing adipose tissue insulin resistance. The adipose tissue does not respond to insulin the way it should. Therefore, it's releasing more fatty acids to the circulation that get to the liver. And other studies, this is a very nice multiple labeled study from Elizabeth Park's group, and they've shown clearly that the majority of fat in the liver and in the peripheral triglycerides is actually coming from these, from the adipose tissue derived free fatty acids. So that's the main source for the fat accumulation. What about the other pathway, the de novo lipogenesis? And you'll hear a lot more about it from my colleague, but if we just focus on here again, another multi-labeled study, it's markedly increased in patients with fatty liver disease, about double than what you see in others. So we have both excess delivery of fat to the liver and excess production of fat from carbohydrate precursors inside the liver. Now, taking a step back and thinking about insulin signaling, as I said, insulin is the hormone of energy presence, of energy excess. And the normal action of insulin on the liver does more than one thing. It tells the liver there's no need for endogenous glucose production, don't release energy, store energy. And it also tells the liver, take free fatty acids and don't use them for energy, store them as triglycerides. So if we're talking about liver insulin resistance, we should expect decrease in fat accumulation and not an increase in fat accumulation. And one of the theories, it's not universally, I think, accepted, but it's gaining more ground, is that what we have is actually a selective insulin resistance. The pathway that controls carbohydrate metabolism is resistant. So the liver, despite the insulin delivery, is pouring more glucose, is generating less glycogen, is worsening through those feedback loops, the hyperinsulinemia. But that the pathway towards fat formation, towards triglyceride formation is intact. So there's actually excess insulin, and this is driving more and more formation of both de novo lipogenesis and generation of triglycerides. And this was nicely shown in a human study which looked at some rare patients, and there are some NIH patients included in this study as well, with mutations in some of the insulin signaling pathway. So when you look at people who have a mutation in the insulin receptor, they're completely resistant to insulin. There are some bypasses, but essentially they're resistant to insulin. They cannot respond to insulin. And although they have very severe diabetes and they require huge doses of insulin, they never develop fatty liver because their liver is completely resistant. When you look at people with a mutation in AKT, which is down the path, separate after the fork, essentially separating only down the path towards the carbohydrate metabolism, these patients have severe diabetes, but also have excess fat in the liver. So they behave a little bit more like the obese non-alcoholic fatty liver patients. Looking at fat metabolism the other way, what happens to these free fatty acids? So we said one option is to beta oxidize them, to utilize them for energy or for fuel. And the truth of the matter is that the data is unclear. Some papers suggest that there is an increase in beta oxidation in patients with fatty liver, but it's not sufficient to handle the excess load. Some studies suggest that there's actually a decrease. We're actually looking at this from another perspective with Gigi Naguib, who's sitting here. And we're using a breath test study for this. We're using a fatty acid that's labeled with a non-radioactive isotope, non-radioactive carbon. And we're using this device that measures in the breath the ratio between the labeled CO2 and non-labeled CO2 at real time. And we're just comparing, for now, we're just comparing fatty liver patients with controls. And we get, this is the typical curve that we'll get after, during the 6 hours of the test. You see, after the patients ingest the meal, and there's a gradual appearance of the label in the expired air when the palmitate is metabolized, oxidized, and some of it goes out as CO2. And the timing of the peak, so we look at two things. We look at the peak of the curve, how fast they actually do it, and we also look at the area under the curve, which tells us essentially how much of the label we're getting, how much of it did they oxidize. And we see that the timing of the peak does not differ between healthy controls, including some very nice and postdocs, and patients with fatty liver. But on the other hand, when you look at the area under the curve, and these are, I think we have like 10 and 4 in the group, and it's already very significant, it's double. So the patients with fatty liver disease actually metabolize about half as much as their healthy controls in terms of palmitate. Remember, this is not a liver test. This is an overall a whole body, so there are other organs that play a role. But as a crude measure and something that's very easy to do, it's very interesting, we think. I haven't touched on the VLDL secretion, but in general, to summarize this part, there's excess delivery of fat to the liver because the adipose tissue is resistant to insulin. There's excess formation of fat in the liver because the liver is resistant to insulin. And there's excess glucose, which is actually driving that as well because you have more products to metabolize. We're not really sure if there's an increase or decrease in beta oxidation, but whatever it is, it's not sufficient to handle the load and even maybe actually cause some toxicity because of incomplete oxidation. And there is an increase in VLDL secretion data that I didn't show you here, but it's definitely not enough to compensate. So eventually what happens is that we accumulate those free fatty acids and they are converted to triglycerides. So that's okay, that's about fatty liver, but why should we actually have the itis, the steatohepatitis? Why should we develop damage from that? So initially, and this is wrong, I should put an X on this. Initially, the model was a two-hit model. You have insulin resistance, you have accumulation of fat in the liver. The fat in the liver causes the liver to even be more insulin resistant. And then comes a second hit, and this could be cytokines or reactive oxygen species or LPS. Pick your favorite buzzword or whatever is the popular topic of the year, and that causes the damage. So there are some pieces of information that contradict this and suggest that this is not the case. First, triglycerides are not bad. Well, they're not good for you, but they're not bad for your liver. And this is a classic example from Anna May Deal's lab. And what they've done, they've actually blocked the key enzyme that DGAT2, which is the diacylglycerol acyltransferase. This is the enzymes that the key enzyme in converting 3 fatty acids with glycerol to triglyceride. So this is the enzyme responsible for generating fat from 3 fatty acids. You knock it down, and you feed the animals a diet that produces fatty liver disease. So these are the control animals. These are animals that they've diet-induced fatty liver disease. They have an increase in their liver fat, and they have an increase in liver enzymes because of injury. And when you knock down these animals' ability to generate triglycerides, you don't get fatty liver because they can't. They just can't make the triglycerides. What you do get is tremendous amount of damage. So the fat here is not the cause of damage. It actually may be protective. It may be a way to shunt other toxic things into a molecule that's relatively inert. Do the triglycerides themselves drive insulin resistance in the liver? It's unclear, but there's some data from Sam Klein's group that suggests they don't. And what they did is elegantly, they used people with a genetic defect, people with familial beta lipoproteinemia. They cannot, or they have a defect in secretion of triglycerides from the liver. So they accumulate triglycerides in the liver, not because of the classic fatty liver, but because they can't throw them out. And they actually do not have significant insulin resistance, or they're comparable to people with the same weight, even if they're overweight, but they don't have insulin resistance in the liver similar to people with fatty liver disease. So again, triglycerides don't seem to be injurious, and triglycerides don't seem to promote insulin resistance. And finally, clinically, the clinicians here know that most of our patients with bad steatohepatitis don't necessarily have a lot of fat in their liver. Actually, those that have a lot of fat in the liver a lot of times don't have anything else. So there seems to be a dissociation between the two phenomena. So then what does cause the problem? And again, this will be discussed further later, but the concept we think about now is of lipotoxicity. Is that actually metabolites of either the fatty acids themselves or their metabolites like ceramides or diacylglycerol or other derivatives are the ones that are accumulating and causing the damage. So essentially, forming the fat in the liver may actually be good for you. It's a way to protect you from those other pathways that can cause damage. There are some data on dietary factors, and an interesting concept related to beta oxidation is that what we may be having an overloading of the mitochondrial capacity. So the mitochondria are working to beta oxidize these fatty acids. They can't handle the load, and then you get partial oxidation and you get reactive oxygen species, which drive to oxidative stress, ER stress, and again, a lot of buzzwords, but essentially, these are all mediators of damage. So there are other mediators that I won't really go into in detail just because we don't have the time to discuss everything, but cholesterol also plays an important role in the disease, although we tend to ignore it. The gut-liver axis and the microbiome are fascinating, some fascinating data there that suggests that they play a role. Just one word about genetics. They're probably not as strong as we'd like them to be. This is, mainly I put it there because I wrote it, so I thought I might as well put the graph there. But one of the strongest hits is a gene called PNPLA3, or the protein is called adiponutrin. And we and other people have shown that it's a mutation in it is highly associated with the degree of injury and the degree of fat in these patients. And it's interesting that the degree of injury is independent of the amount of fat, again, suggesting that there's a discrepancy. Irrespective of how much fat you have in your liver, you'll get worse injury if you have the risk allele. Is this the major player? Now, modeling data suggests that this explains about 2 to 3% of the variance in the population. So most of it is probably not genetic, although there is a serious thing that we haven't been able to answer yet, and this is the racial difference. Hispanics are very prone to fatty liver disease. African Americans seem to be protected from fatty liver disease. It's very rare to find an African American with NAFLD, and we haven't really found a good explanation for it. I clicked too much, and just talking about, we'll finish with some of data on what's available now for treatment. So I'll cover a few strategies. Targeting lifestyle. We'll talk about the insulin pathway. We'll talk about the bile acids and FXR pathway, and about the antioxidants. So diet and physical activity. I sound like a broken record whenever I talk to my patients, but that's the mainstay of treatment. Like all of the other components of metabolic syndrome, diet and physical activity will help with diabetes, with hypertension, with obesity, with hypercholesterolemia. Bariatric weight loss surgery is an excellent therapy, and this has been shown in multiple studies to promote histological response. You can actually get the liver to look better. This is some data that we collect from, basically this is a prelude to another study, but where we have 12 weeks of lifestyle intervention. Essentially, we have the patients come in every 2 weeks to meet a nutritionist, and we give them pedometers and ask them, walk more and write down. That's all. We don't put them on a treadmill. We don't give them the food or anything like that. And this is, you can clearly see that as their BMI decreases after 12 weeks, so does their liver fat, and so do their liver enzymes. And it's very strong correlation, very convincing. We reach, after 12 weeks, a third of them have normal amount of fat in their liver. A third of them have a normal amount of, a normal level of ALT through very strict criteria. And actually about 15 to 20% of them on a liver biopsy don't have fatty liver diseases anymore. People that we know had fatty liver disease, after 12 weeks, they have a near normal or a normal liver biopsy. What's the problem? It's unsustainable. Like many studies of weight loss in obesity or in other diseases, this cannot be kept for long. And seriously, we do it for 12 weeks, and then we just see them every 8 weeks with the nutritionist, and they regain their weight. This is just their body weight, and weight continues to go up, even though they're still in a clinical trial, even though they're still seeing a nutritionist. And this has been shown in multiple studies. This is very difficult to sustain. So then we have to look into medications to help us. And I think the first and probably the most successful path so far has been to look into insulin sensitizers, mainly because they've been approved for diabetes. So it's already there. We can just let's throw another diabetes drug at our patients. And a lot of these work through the PPAR pathway. I won't go into it in too much detail because this is not the topic, but the PPARs are nuclear receptors that regulate a lot of the energy metabolism in the body. Their ligands are either fatty acids or their derivatives, and they're transcriptional regulators with RXR through a shared motif. There are 3 PPARs, alpha, gamma, and delta. They differ in action they all detect the same motif. They differ in action by the ligand, by their tissue expression, and the result is that PPAR alpha is mainly a fasting signal. Where's our fasting guy? Mainly a fasting signal driving fatty acid oxidation, and where PPAR gamma is working predominantly in the adipose tissue to improve insulin sensitivity, but also in the liver. And PPAR delta is responsible for other things, but including peripheral insulin sensitivity. Pioglitazone is, I think, the main medication from this group that's worth mentioning. It's clinically used for diabetes. It's a PPAR gamma agonist. It was studied in multiple studies for fatty liver disease. This is an example from the PIVENS trial, which NADDK funded. And if you look just at the blue line comparing to placebo, this was for 2 years, and you see that the liver enzymes decreased nicely, about 35 units of ALT through treatment, and they improved their insulin sensitivity. However, they gained about 5 kilograms of weight, and that's a known side effect or effect of the PPAR gamma agonists. There have been meta-analysis on it, and the bottom line is PPAR gamma agonists work for fatty liver. They improve liver histology, they're beneficial, they're helpful, they're safe in patients with liver disease. However, we're talking about histological improvement only in about 40% of patients, depends on the study. They gain weight, which they don't like it, and there are some safety concerns that are class effect and probably are not that concerning, but they're there and people don't like taking them. A new member from this class is elafibranor that was published about a few months ago, a phase 2 study. This is a PPAR alpha delta dual agonist. So it acts on those 2 pathways at once. Has shown very promising data from rodent studies. And this was 280 patients randomized to placebo in 2 different doses for 1 year. And they had the very ambitious endpoint, the complete histological resolution of NASH. And they failed to meet their very ambitious endpoint. They tried to justify, the graph is mine, it's not there, but I used their data. And they tried to justify it by saying that it was beneficial in the patients with severe disease, and they included too many patients with mild disease. Let's say it this way. But if you look at their response criteria, using the same criteria that the study, the vitamin E, the pioglitazone study used, they have the same numbers, 48% responders versus 21% in placebo, so looks really about the same. And finally, along the same line of insulin sensitizer, I want to mention liraglutide, which is a GLP-1 agonist, or a GLP-1 mimic. That's a better term for it. GLP-1 is a gut-derived hormone that is secreted from the intestine when we eat, actually even before the food gets there, probably. And it acts as it improves insulin sensitivity both by action on the pancreas to increase insulin and decrease glucagon. And also it has activity on the periphery, mainly the liver, and decreases appetite as well through action on the central nervous system. So it's also associated with weight loss. There have been some retrospective analysis of studies, and this is approved for diabetes, and people have analyzed either this or other medications from the same class, and a lot of them show benefit in diabetic patients in retrospective analysis. You take the diabetic patients, you look for those that had elevated liver enzymes, you look at what happened to their liver enzymes in the study. Oh, it went down, that's nice. But this is actually the only real study. It was just published a few weeks ago in the Lancet. It's actually a small trial, 52 patients. Were randomized to liraglutide or placebo for 48 weeks, and they've had 40% resolution of NASH. Does the 40% number sound familiar? We're repeating ourselves. And this was significantly better than placebo, which is actually surprising success for a small study. And the liver enzymes decreased nicely by about 30 to 40. Again, a number that sounds familiar from previous graphs. And they also lost weight, and it's actually not clear whether the benefit is independent of the weight loss. They don't have a large enough number to say whether this is just a side benefit of losing weight, in which case you may have other ways to treat it, or is this directly related to liraglutide? But this is something that we'll probably hear about more in larger studies. Bile acids, I want to talk a little bit about the bile acid pathway that I haven't mentioned before. So those of us who went to medical school or biology 101 learned that bile acids are secreted from the liver, and their role is to allow for absorption of fat from the meal. They form micelles, they allow the water insoluble fat to become water soluble and then be absorbed. The majority of the bile acids are reabsorbed at the terminal ileum at the far end of the small bowel, and then get recirculated. So most of the bile acids that are secreted from the liver get back to the liver, and there's a whole pathway of interaction with the gut bacteria. There is a very interesting nuclear receptor called FXR, which, and again, if some of you were present at our visiting professor's talk last week, that was actually the topic of a lot of the discussion. And FXR is acting as an intracellular sensor of bile acid levels in a way. Bile acids are the ligand, and especially in the liver, when FXR is activated by bile acids, it will transcriptionally regulate and decrease CYP7A1, which is the rate-limiting enzyme in formation of new bile acids. So if you have bile acids, you don't need to make more. You can downregulate that pathway. And it will also upregulate the transporters that secrete bile acids out of the liver. So in a way, this, in a simplistic explanation, this is something that controls or stabilizes bile. It's a feedback loop that stabilizes intracellular bile acid levels. FXR also acts in the terminal ileum where the bile acids are absorbed, and actually acts both on the transporters and on a hormone, on a gut-derived hormone called FGF19 that then can act directly on the liver and have an insulin-like effect. However, it was noted that FXR activation does a lot more than that and has a strong metabolic role. It affects lipid metabolism. It sensitizes, FXR activation sensitizes to insulin, decreases de novo lipogenesis, increases fatty acid oxidation. So this could actually be beneficial. And lo and behold, a company actually came up and made obeticholic acid, which is a synthetic modified FXR agonist, and very potent, very promising results in preclinical models and early clinical trials. And the NASH CRN network that previously did the pioglitazone study went ahead and did the Flynn trial with this medication, 72 weeks, double-blind, randomized, placebo-controlled, 200 and something subjects. All of the studies are the same. It's very easy. And the result, yes, 45% versus 21%, exactly the same numbers. Liver enzymes decreased by 30 to 40, exactly the same numbers. And a very favorable profile in terms of enzymatic response, and also, this is the first improvement to show some improvement, first intervention to show improvement in fibrosis, which is something we really like, although I don't know that the data is that convincing that it's that strong. But this actually surprisingly worsened insulin resistance. This worsened cholesterol profile, and about a quarter of the patients itched so much that some of them had to stop. So, and when you look at resolution of fatty liver disease, not improvement, it's only 20%. So this is actually started a phase 3 study. This is the first phase 3 trial in fatty liver disease, but whether this will actually catch as a good treatment, I'm not convinced. Finally, I want to talk about antioxidants. So up until now, we talked about things that are related to the accumulation of fat and injurious metabolites. But another approach can say, okay, you know what? I can't deal with the fat. you have fat, stay with it. You have the toxic metabolites, stay with it. But if the damage is driven by reactive oxygen species, let's just treat that. We'll just take care of the oxidative stress with antioxidants, and that can solve the problem. So the best target for this is vitamin E because it's a lipid-soluble antioxidant and because it's widely used and widely available. And the same study that I showed you earlier with pioglitazone actually had an arm with vitamin E. I think they used that arm just to calm those vitamin people down, that just get rid of them. And surprisingly, 43% had a histological response versus 20% in the placebo arm. It actually did better than pioglitazone. And if you look here, you've seen this graph before, but the green curve is the vitamin E liver enzyme response. Again, it dropped down by the 30-40 magic number. It did not impact insulin resistance because it's not supposed to. It's supposed to be way downstream of the path. It did not cause weight gain because it's unrelated. So it looks pretty good. One interesting thing that's been missed here is that both in that study, in the pediatric study that was done at more or less at the same time, this was actually associated with a decrease in the amount of liver fat. And if you think about it, it doesn't make sense. It wasn't supposed to. This was supposed to be downstream of the liver fat, of the injury from liver fat. It shouldn't have affected liver fat. Why does it do it? No good answer, but I have a study, so hopefully in a few years I'll be able to tell you. This is some clinical data from that study, the study is actually going to look into molecular mechanisms, but just some of the clinical results that we have, we replicated it. After 24 weeks of treatment with vitamin E in patients with fatty liver disease, we normalized their ALT in 50%, we decreased their liver fat in 80% and normalized it in 20%. And we actually have a liver biopsy very early on at 4 weeks. And very early, within a month of treatment, 60% of them show histological improvement. So it definitely works, although we can argue what the mechanisms are. We're looking at different doses, and it looks as if there's a trend, but it's too early to say anything about significance. And just to finish more or less by comparing the different options that we have. So I've spoken about all of these, and most of them ranging around the 40%. The best treatment is lifestyle, but again, the issue is whether it's sustainable. Resolution of NASH, lower numbers as well. And I think that we can summarize by talking about the challenges that as clinicians we have in studies of fatty liver disease. We're dependent on liver biopsies because we actually don't have good enough markers that would be acceptable for treatment trials. We're actually, and again, because we're dependent on liver biopsies, we're treating histology, which does not necessarily imply that we got their livers to look better, but does that mean that they will die less of liver disease or of cardiovascular mortality? We don't know. We don't have the 20-year-long studies to show that, and it's going to take a long time until we actually know. And we don't actually have studies that treat patients continuously for 20 years or so. We don't have any data from phase 3 trials. We only have one study that started. And finally, humans are different than mice. A lot of the things that are promising in animals have failed or have been not as successful in the humans. We'll finish with John, the patient I told you about before. So we biopsied him. He had advanced disease, actually almost cirrhosis. He went through the lifestyle intervention. The blue line is his weight. He lost weight nicely. His liver enzymes improved. His liver fat content actually normalized. Then we put him on vitamin E. He actually improved some more with the vitamin E within those 4 weeks. At week 24, he had no fat in his liver. This is by MRI, but he had 0.06%, nothing in his liver, and his enzymes were the normalest they'll ever be. And then while on treatment, he started to regain weight. He regained fat. He regained enzymes. So the weight is actually probably the most important player, irrespective of what medication we can give them. Wrong side. And with that, I'd like to thank, and I highlighted the people whose data I've shown you, but the members of my lab, all of the members from the clinical section at the liver diseases branch under the mentorship, guidance, leadership of Jake Liang and Jay Hufnagel, some of our collaborators from nutrition, from imaging, pathology, and some people that collaborate with us, and of course our patients that help us. This is our group, and I hope you have a better Ides of March. Thank you.
Thank you. That was wonderful. So please, we have time for some questions. Sort of keep them short. Use the microphone.
Very nice, Yaron. Just a question about this notion you brought up regarding the, for example, people of African descent not having much. How many genome-wide studies or any other indicators have found candidate genes that could explain those differences that you're seeing among these different types?
So the biggest hit in genome-wide studies has always been the PNPLA3 that I've shown. And it is actually, there's a difference in prevalence between populations. But the risk allele is present, if I remember correctly, in 17% of African Americans versus 21 or 22% of Caucasians. It's not enough to explain the difference.
On your breath test, does it make any difference if the human being is fed or starved, fasting?
I'm sure it does. We started the fast after an overnight fast to sort of, and we feed them a defined meal, so we can control it. It's much more difficult to do something like that after a meal because then there are also issues with absorption, with delaying of gastric emptying by the previous meal, and all of that. I should mention there's very interesting data with labeling studies that suggest that maybe we should be looking at them after the next meal, because there's some data that suggests that a lot of the fat from meals actually stays in the intestinal cells and only secreted from them after the next meal. So that as if there's one meal lag in secretion. Of course, we see some of it because otherwise we wouldn't see any label, but we also have to be practical in the clinical trials.
Okay, so we have one other.
Just in a follow-up to the C13 experiment, was the respiratory quotient the same at the time you did that? So was the rate of fat oxidation the same in both groups? Was that so the question is, are you seeing a difference in fuel partitioning between carbohydrate and fat and what's being burned? And that would be really interesting if you see, for example, the RQ is no different, so you're burning the same fraction of carbohydrate fat, yet you're seeing the recently eaten palmitate is lower in the NAFLD.
So the short answer is that I don't know. The longer answer is I don't know yet. And the even longer answer is that a lot of these patients are actually participating almost simultaneously in another study that we have, which is performed at the metabolic chamber with almost the same challenge meal. It's a different trial, but it's not exactly the same, but it's more or less. So we will have RQ values, not from the same moment, but on most of these same subjects. We actually have the same controls and fatty liver, so we should be able to answer that question.
So prior to 1990, when alcoholic fatty liver was the vogue, numerous studies showed that the basic mechanism ultimately related to acetaldehyde and the production of reactive oxygen species and so forth and so on. So my question is, what happens to someone who has NAFL, who also has a few beers once in a while? Are you really effective in screening all alcohol intake from these patients? And what would the effect be if you have a PLP, for example, a mutation or some other environmental sort of business, pre-diabetic, all the things that make a syndrome? What happens if you add a little bit of alcohol to that?
So the data is somewhat conflicting, but I think at least at the level of epidemiology, there's a suggestion that low-level drinking or moderate drinking may actually be beneficial for these patients, the same as it's been suggested for cardiovascular disease. And again, whether this is wine versus beer, whether all alcohol is the same, whether it's the alcohol or other things in the drink, I don't know. How good are we at screening? As best as it gets. There's actually no clear definition of what non-alcoholic mean. Most of them are not complete abstainers, teetotalers that never drink. It's acceptable, at least in the West, what usually is acceptable is up to 2 drinks a day, maybe 3 drinks for a man, but it varies from study to study, from guideline to guideline.
So it's not like Caesar's wife.
Nope.
Rosanna, I think.
Yeah. Okay.
So, Yaron, so why do you think that we get that hit, that 40% hit? Every single study has been done only just with one drug. Do you think because we still haven't found the right mechanism, or maybe because you need multiple mechanisms to see the full resolution of NASH?
So the answer is that I don't know. And I think an even more important question, or the same question can be asked from the other side. Are non-responders just non-responders, or are they non-responders to a specific medication? So is the person who's responding to pioglitazone, will they also respond, or can we tailor one medication to one person or not? I don't know. It's a study that I'd love to do.
Yeah, stratification of these patients are very important because it's not only one disease; there are multiple diseases.
Probably.
Yeah.
Okay. So, Rosanna.
Thank you. So first of all, Wynn, thank you so much for inviting me here today. And I want to say a few words just to clarify what Wynn said. So yes, I come from academia. I'm an MD PhD trained in Wynn's department. I'm not an administrator. Okay, so I just want to make that very clear. Actually, since I was in medical school, I wanted to be part of translation. I wanted to see drugs coming from the bench to the patients. And actually, I believe that's what got me into medical school, because Wynn liked my sentence I said from bench to patient or something like that. At that time, this was not something that people talked about. And I decided actually while I was in my PhD that I wanted to go into biotech, that I did not want to stay in academia. So actually it was very deliberate. And the reason for that is because I wanted to be part of the translation community. And I have to say that only took me 20 years, but there's always hope. And that now actually at Nimbus, we have been able to do that, or we're on the way of doing it. So what I want to talk to you about today is the work that we have been doing in generating inhibitors against acetyl-CoA carboxylase, which basically is the key enzyme involved in fatty acid synthesis. So therefore, the title of my talk, which is Trimming the Fat, Inhibiting Acetyl-CoA Carboxylase to Treat Non-Alcoholic Steatohepatitis. So Yaron actually gave a fantastic talk introducing the disease, and there are no words for it. NASH is going to be probably the biggest epidemic of this century. It's going to surpass HCV for liver transplantation. We have to figure out a way to treat these patients. The issue is, it is a sequela of obesity. Actually, I think that a very good percentage of NASH patients are obese. Not all of them are obese, and as we're talking about right now, there is a lot of heterogeneity in the disease, but a great percentage is obese. And actually, lifestyle modification does work. It is the best way to treat NASH. But the problem is, as with obesity, is not something that is sustainable. For some reason, we love to eat, and it's not a sustainable thing. So let's talk about the numbers. Why is that such a big problem? So as Yaron said, there's about 30% of adults and about 10% of children now in the United States that have NAFL, okay? So if you translate that in numbers, more or less, that's about 80 million people. 80 million people just in the United States, okay? And then from that, 10 to 30% will develop NASH, which is characterized by inflammation, apoptosis, and fibrosis. So that's about 16 million people. Again, 10% of those children, 1.6 million children with the risk of developing cirrhosis. And then when you continue the math, 20 to 30% will develop advanced NASH with advanced cirrhosis, and then a percentage of that will end up in liver transplantation. There's a cardiovascular risk. Right now, the estimate is about by 2025 that we're going to be spending $35 billion in taking care of this problem. So we need to do something now. And we already agreed that lifestyle modification does not work. Yaron introduced the lipotoxicity injury hypothesis. As he mentioned before, there was a two-hit hypothesis of what causes NASH. I think that now the community is coming to agreement that probably it is not the fat itself, but it's actually the accumulation of free fatty acids in the liver that lead to the accumulation of lipotoxic metabolites that induce your stress and necrosis and fibrosis. And how do you get that? How do you get these free fatty acids? We talked about diet, and the lack of exercise. You have excess carbohydrates leading to de novo lipogenesis. And of course, the counterpart of that is insulin resistance. And I'm going to take you back 40 years ago. when we actually start understanding more about cardiovascular disease and understanding more that cholesterol was a culprit of cardiovascular disease and how we're going to treat cholesterol. And the first thing we told patients is stop eating eggs because eggs are bad for you, decrease your cholesterol intake. That didn't solve the problem at all. And then we developed a drug that most of us use, statins, that are HMG-CoA reductase inhibitors. So if you can't reduce the intake, can you actually improve the condition by blocking the endogenous production of fatty acids? So here comes my favorite enzyme, acetyl-CoA carboxylase. And what acetyl carboxylase does is the rate-limiting step in the production of de novo fatty acid synthesis, and also it is an inhibitor of beta oxidation. So basically, if you inhibit ACC, what you're going to do is decrease de novo lipogenesis and increase beta oxidation. So let me tell you a little bit more about it. There are two isoforms of ACC, ACC1 and ACC2. ACC1 is cytosolic, mostly present in lipogenic tissues like the liver. And ACC2 is actually present in the outer membrane of the mitochondria and is present in oxidative tissues like muscle. But the reality is that ACC1 and ACC2 are in all tissues, and if you block one and not the other, they will find a way to compensate for each other because it's an essential enzyme. What they do is convert acetyl-CoA into malonyl-CoA, which is the first building block of fatty acids. And also what is important to know about malonyl-CoA is that malonyl-CoA inhibits CPT1, which is the transporter involved in fatty acid translocation into mitochondria. So basically, by doing that, blocking beta oxidation. Very smart. So if you inhibit ACC1 and ACC2 simultaneously, what you're going to get is a decrease in fatty acid synthesis with a concomitant increase in beta oxidation. If you think about that, that's what happens when you exercise. That's exactly what happens when you exercise. And by the way, ACC actually is regulated by AMP kinase, which is actually going to become important in a minute. So we talk about ACC. So what are kind of the proof points that we have that actually blocking ACC could improve NASH? So this paper actually was published now a couple of years ago, and it's one of the most elegant papers I have ever read. And what they did is that they express constitutively active ACC. And the way they do that is that they do point mutations on the serine residue that's phosphorylated by AMP kinase. So AMP kinase can no longer phosphorylate ACC, and they do that for both ACC1 and ACC2. They generate these transgenic mice, and what do these mice have? NASH. So they have increased hepatic triglycerides, they have increased fibrosis, and decreased insulin sensitivity. And also, what I don't show here, they also have increased inflammation, and I'm going to talk a little bit more about the inflammatory part of NASH, which is also very important. The counterpart of it is if you use, for example, RNA-based technologies to knock down the enzyme, and you knock down ACC1 and ACC2, you increase fatty acid oxidation and decrease fatty acid synthesis. I just want to stop here for a moment and pause because there is a lot of controversial data using knockouts of ACC1 in the liver, ACC1 and 2 in the liver, ACC2 in the muscle. It's very confusing. It's very dependent on the backgrounds that you're using for the mice. I would refrain from making too many conclusions from that. But in the paper that's published right now with ACC1 to knockout in the liver, they claim to see an increase in liver triglycerides in the liver, which very much could be due to the mechanism that you just talked about, the lipolysis and basically fatty acids getting into the liver. Something to remember, but that's not what we see when we pharmacologically inhibit ACC. So what does ACC do? I said it's important for the conversion of acetyl-CoA into malonyl-CoA. And the only reason I'm showing you this slide is because this is a two-step reaction. The enzyme is a multi-domain enzyme. It has a biotin carboxylase domain, a biotin carrier protein domain, and a transcarboxylase domain. And what this biotin carrier protein domain does, it basically swings biotin from one side of the enzyme to the other side of the enzyme. And the first steps occur at the biotin carboxylase domain, where there's a carbonyl that's put into the biotin, and then it's transferred to the transcarboxylase domain, and that carboxylase domain actually put that carboxylates acetyl-CoA, making malonyl-CoA out of it. So if you look at this enzyme, a priori I said, oh, if I want to inhibit this enzyme, I can either go after the carboxyltransferase domain or the biotin carboxylase domain. Most companies actually went after the carboxyltransferase domain because they couldn't find a way to inhibit the biotin carboxylase domain. However, that's what we did. We took a very orthogonal approach and went after the biotin carboxylase domain because on the biotin carboxylase domain, what you have is an allosteric site that actually regulates the physiological inhibition of the enzyme by phosphorylation induced by MP kinase. So this is actually has just been published recently. Leon Tong, who is a collaborator and has been studying acyl-CoA carboxylase for a long time, what he shows is that when the BC domain is phosphorylated, the pointer is not working, I think, because, let's see. No. So when, I don't know if you can see it, but basically when the BC domain of ACC is phosphorylated, there's the phosphate there, it creates a bump that doesn't let the carboxyltransferase domain work and doesn't also let the BC domains or the holoenzyme to dimerize. So it blocks the activity of the enzyme. And when the enzyme becomes dephosphorylated, there is a conformational change that allows for the dimerization and activation of the enzyme that only occurs in the eukaryotic enzyme. There is a fungicide called Sorofen that binds to that same allosteric site and inhibits the enzyme. So this is known for 10 years. And people have been trying to target that Sorofen site, but failed to do that. And I'm going to show you why in a minute. So this is just depicting the phosphorylation of the A domain of the BC domain tail, and basically that phosphate, now it's binding to this residue here and basically blocking the dimerization of the protein. And what we do, either Sorofen or our inhibitor, it sits exactly on that pocket. And by the way, it coordinates with the same residues that the phosphate coordinates. So what our inhibitor is doing is mimicking the physiological inhibition of ACC, okay? Which is very different from all other inhibitors. So this is actually the target site. And what you see in green, that's represented by the sticks and the balls, is Sorofen. And you can see, even if you don't know any chemistry, like me, it's like basically it's this very big macrocycle that sits as a cork on that particular site. But and the spheres, the red spheres, represent high energy waters. These are waters that are trapped in the site and are not happy. So basically the theory goes that if you can actually displace those waters, you're going to increase the affinity of the ligand for the receptor. And we work with a company called Schrödinger that does computational chemistry, and they are our partners, and they characterized those waters, and that's what we did. We went after those waters and basically generated now a molecule that is incredibly potent. So just making a long story short, this is NGI-976. This molecule is in the clinic. We finished phase 1 studies. We're phase 2 ready. We're presenting actually our clinical data at EASL in Barcelona in a month. But today I'm only going to talk about the preclinical data of this molecule. But that molecule basically binds to that same site, precludes the dimerization of the protein. It's very active. It's about single-digit nanomolar against the enzyme. And that translates very nicely with potency in both the cellular assays and in the animal models. And I want to call your attention to this inhibition of hepatic fatty acid synthesis. So what we do there is we infuse the animals with C13 or C14 acetate in that case, and then we look at the production of triglycerides or palmitate, and we do exactly the same thing in humans. And what I'm going to tell you is that the dose that we're using in the animals to block the production of palmitate is basically the same dose that we're using the rats to block the production of palmitate is the same dose that translated into the humans to do that. So we can now calculate exactly what the effective dose is. To produce NASH, you need 3 things. You need steatosis. Steatosis, as Yoran said, is not enough. You need inflammation and apoptosis, and you need fibrosis. And the only way that you're going to be able to treat NASH is if you can actually block each one of these pathogenic processes. Just blocking one or the other may not be sufficient. So we took an approach to separate the models and took a mechanistic models to study only steatosis or study inflammation and fibrosis. And what I'm showing here is that we have a very nice reduction in the production of myelonucleae both in the liver and the muscle. That's dose responsive. And that translates very nice with decreased fatty acid synthesis in the liver, as I talked about before, and also an increase in fatty acid oxidation, which is also dose responsive. We did several models. We did 14-day DIO studies, 28-day DIO studies, high fat, high sucrose. We did a ZDF study. And in all these models, every single one of them, the most impressive data point we got was a decrease in liver-free fatty acids, a decrease in liver triglycerides, decrease in plasma triglycerides. And very different from the findings from OCA, we also see a decrease in cholesterol that actually translated into humans. So our human subjects have decreased cholesterol, not increased cholesterol. Then the second piece, so we have shown that we can decrease steatosis in the liver. Now, can we decrease inflammation and fibrosis? And we decided to take a very different approach in terms that we wanted to separate steatosis from inflammation and fibrosis, because one of the criticisms that we got was, oh, everything you're doing is because you're decreasing fat or decreasing the body weight or whatever. And we wanted to say, no, it's a direct impact in inflammation and fibrosis. So we used this diethylnitrosamine rat model that recapitulates the histological changes seen in NASH. So as you can see, 8 weeks of injecting DN into these animals, you have early fibrosis, you see lobular inflammation, hepatocyte ballooning. And about 12 weeks, you see increased fibrosis or bridging fibrosis. The lobular inflammation, hepatocyte ballooning is there. And by 18 weeks, actually, you see cirrhosis, and you also see hepatocellular carcinoma. So this goes through all the changes seen during NASH all the way to cancer. And what you can see here is that when these animals are treated with our inhibitor, we see a decrease in the production of smooth muscle actin in the liver, and that's translated not only at the protein level, but also at the gene expression level, basically showing that we're blocking stellate cell activation. And actually data that I'm not showing here is we have that data also in vitro showing that's a direct impact of ACC inhibition on stellate cells. And the other piece of it is that we also looked at inflammatory markers and other fibrosis markers. And what I can tell you is that DN induced all these inflammation markers, for example, like MCP1 and TNF-alpha and LIF. And then basically we see a dose-responsive reduction in all the inflammation markers, and we also see a dose-responsive reduction in fibrosis. That piqued our interest because why are we seeing such an impact in inflammation? And one thing that I want to bring up is Th17 biology. I don't know if you guys are familiar with Th17s, but they're effector cells, inflammatory cells, and they have actually been implicated in NASH, in the progression of NASH. So in animal models of NASH, if you block Th17, you can have all the fat in the liver, you can have all the fatty acid accumulation in the liver, you cannot progress to NASH. And then when you look into humans, there is an increase. This is all phenomenology right now, but there's a huge increase in hepatic Th17 cells and RN gamma T and IL-17 and IL-23. So actually there is a whole body of work that's now exploring the role of Th17 in the liver. And in addition to that, there are 2 papers that came out recently showing that ACC activity is key for Th17 function. So in the first paper, what they show is that if you inhibit ACC, you suppress Th17 differentiation and actually trigger Treg. So Treg are the suppressor cells. Th17s are the bad actors, and Tregs are considered the good actors. And then in cell, basically what they show is that ACC activity is required for the biosynthesis of the endogenous ligand of our gamma T, which is required for Th17 differentiation. And what we show here is actually we have a beautiful dose-responsive decrease in Th17 differentiation in vitro, and what we're not showing here is that we have an increase in Treg. We actually now have done models of EAE, which is a Th17 model, and we completely block EAE in these animals, basically showing that we're blocking Th17 activity in vivo as well, and we're translating this work now into liver. So what we know today is that 976 or other ACC inhibitors of the same class not only block the accumulation of fat or fatty acids in the liver, but they also block inflammation in the liver, and they also block fibrosis in the liver. basically impacting all the three pathways that are required for the pathogenesis of NASH. So in summary, we have the first-in-class allosteric ACC1 and 2 inhibitor, and we're very excited about that, and that basically mimics the physiological inhibition of ACC. One thing I forgot to mention is that this is a hepatotropic compound, so one thing that was very important was to target the relevant tissues, and the relevant tissues are the liver and a little bit of the muscle. You don't want it everywhere, and that we impact the key pathogenic processes of NASH. As I mentioned before, we are now phase 2 ready, and we have just been granted fast track designation by the FDA. So we're trying to race with Intercept and GenFit. But what I want to tell you is just to remind you, this is a really important problem, and there's about 48 clinical trials that are currently open for NASH and FLD around the world, mostly in the United States and Europe. And then in terms of all the compounds that are currently in the clinic for NASH, this is basically a summary of all these compounds. And the only thing I want to say is that they are either metabolic modulators, antifibrotics, or anti-inflammatories. There is some work being done with antioxidants, but now they're starting to combine the antioxidants with some of these other drugs. And that's why I asked the question about the combination. So thank you very much. I just want to acknowledge my colleagues that did the work at Nimbus and other places. One thing we do is we develop very strong collaborations with academia. We do a lot of work with academia. So Jerry Herman and Jeremy Greenwood were actually the chemists that discovered and developed the compound. And these are the biologists that took the compound from the computer first into rat and now into man. So when I didn't go from the bench to the bedside, I went from the computer to the bedside. So thank you very much.
Talk a little bit about the FDA registration endpoints for a NASH drug. So
yeah.
With diabetes, we have the A1C, and that turns out to be a great microvascular, not for cardiovascular. So do you think that NASH drug is going to have a hard clinical endpoint?
That is a great question. And there is a lot of discussion about that right now. From the FDA and the EMA. So right now, as Yaron said, the only thing we have are these histological surrogate endpoints. And they are basically the NAS scores, and you have to show a decrease in hepatocyte ballooning. So just showing decrease in fat is not enough anymore. You have to show a decrease in hepatocyte ballooning, and you have to show a decrease in fibrosis. What the whole community is working, really working very hard after is what are the biomarkers, serological biomarkers that we're going to be able to use to predict progression of the disease, but we're not there yet. I mean, there are some candidates, but they're not good enough. Yaron, you want to add anything to that? There's imaging also, but the imaging, it's not.
I'll just speak loudly.
Yeah.
Okay, I have to agree. I mean, there was a shared meeting between the FDA and the Association for Study of Liver Disease 2 or 3 years ago, and the recommendation was actually to go with histology and to go with NASH resolution, which then turned out to be a very difficult endpoint to achieve in the subsequent studies. It's not that GenFit went through this endpoint because they really wanted to. It was because that's what the FDA and AASLD agreed that that's it. And it's really what we'd like to achieve, but it seems that it's going to be very difficult to get there.
So what happened is Intercept, actually, when they started phase 3, they're going after co-primary endpoints. So NASH resolution with no worsening of fibrosis, or resolution of fibrosis with no worsening of NASH. GenFit, now for the phase 3, they're only going with one primary endpoint, which is basically NASH resolution with no worsening of fibrosis. So there is still a lot of discussion. And what the field really hopes is in the next 2-3 years, as we do these phase 3 studies, that we're going to develop better biomarkers, serological biomarkers that are going to be used. I mean, GenFit claims that they now have a microRNA-based biomarker that they can use in their trials, but nobody has seen it, so there's still a lot of skepticism. But one thing I'm going to say is that the FDA and the EMA is working very hard with the community, trying to develop a new way of doing this. And actually, there is the Liver Forum. I don't know if you know about that, but every meeting, every AASLD meeting and EASL meeting, the Liver Forum gets together, and the FDA and the EMA participate, and they have now workshops involving people from industry and academia to try to solve this problem.
So, Rosanna, what's the metabolism of the drug?
It's secreted as a parent by the bile. There is no metabolism to speak of. It's a parent compound secreted by the bile. It basically goes to the liver, and then eventually it just comes out in the bile.
And why does it go to the liver?
Because actually it takes advantage of OATPs. So it's, we designed it. We specifically designed this drug to go to the liver chemically. I mean, very smart chemistry, yeah.
But what the basis of that was?
It's a carboxylic acid. We put a carboxylic acid as
Yeah.
one of the substituents in the ring. And basically that is picked up by OATP.
But there are 5 OATPs.
It's picked up
in the
by
liver.
OATP 1, 2, and 3.
So it's just 1, 2, and 3.
Yeah.
That's amazing.
Yeah, and we have done a lot of work in characterization of the transport mechanism.
So I have two questions, actually.
Yes.
So regarding the OATP transport, do you think that in vivo in humans, you will still see the effect on non-hepatocyte cells? Because it seems like there is, at least it looks as if there's direct effect on T cells and direct effect on ACC in stellate cells. Otherwise, it's difficult to explain. Will that be true also in the human in vivo?
We don't know that. So the only thing I can say is that we have done the in vitro studies both in human Th17 cells and in human stellate cells, and the answer is yes. Okay, and the way that we have done the human stellate cells is very interesting. We're collaborating with the MGH, and he gets a little biopsy when he does bariatric surgery. So we're getting the stellate cells from fat people, so which is great because they are very activated. But we have no way to know that until we go into humans. What I can say about humans is that the experiment that we did in the rat with the C14 acetate, you can do exactly the same experiment in humans. And we have infused humans with C13 acetate, and we have, they were fasting, and then the next day we basically gave them Kool-Aid for 10 hours, fructose every half an hour to induce fatty acid synthesis, and you can increase de novo lipogenesis. And basically we can see also those responsive reduction of de novo lipogenesis in humans. So whatever is happening in the animals, we can now translate into humans. And all these studies are going to be part of our phase 2 and 3 studies.
And another question, if I may, about the two isoenzymes. So I think there's more and more data that suggests that there's partitioning of the metabolism based on cellular location, that you have enzymes that cluster together, and that there's also data that suggests that fatty acids, that the palmitate coming from the adipose tissue will be metabolized differently than the palmitate generated by de novo lipogenesis,
Yes.
even if it's the same enzymes. So is it crucial? Do you know which ACC is involved in which, and whether it's truly crucial for the clinical effect to actually inhibit them both, or whether one of them would be sufficient?
We don't know the answer to that, but we're actually doing studies now, and we're going to collaborate with Christian Metallo to address exactly that. We're starting to do studies in tissue culture and to understand what's the differentiation, but we don't know yet. Yeah, but that's a great question. The reason to basically block both is because there was so much confusion with the knockouts, and we know that one can compensate for the other, that we decided to block both, but be tissue targeted. You see, so to avoid some of the other issues.
Well, thank you both. And this will be a work in progress.
Oh, yeah.
We hope to hear more.
Thank you.